Yes.
So in the first session, we talked about those first two stages being cleavage and gastrulation. Now we are going to talk a little bit more about some of the genes and then therefore proteins that are going to be involved in that third step of neurulation.
That tampo tigla ya ngasuge ko therapeutic rambagana simjir dan todin driven ti ko lai kiche res ra ta tanda bana ngasuge wangsa giten sigdo wangsa ten to be ramba de na ne ane so de ka rigje ta rigje la ten ne ane tizit ko yarwa o tigi cheli kana ro chi be de ta gins.
I wanted us to talk about some of the genes for two reasons. One is that if you remember on day one, I mentioned that my PhD was in genetics. So of course, we could not get through a course without talking a little bit more about genes.
That and the dinner ne so tinga so ge rigzi ko lan de jagi ins ro. Ta ta jinsen chi kar system balaya ani Nima tampula ngarang ngoto chibi kapsa ngarang gi banramba gi lobjante tanda rizi rupi kola che bade chesran de ja yes tin stamala ne rizi kola dus tabde chingoto chigis.
And then the second reason is that I usually hear a lot of questions and a lot of curiosity about how do things actually happen. And so we might say, even like what's the molecular mechanism for what's happening. And this is going to give us a little bit of a peek, at least, as to what's going on with that. What are the molecules involved in this developmental process?
Ta shendachi kare system balaya ane mambo chiki tanda dita kapsita ko litsita chelete kare dawochi yemeng ko la ya dusta ko sinyi tuya ta chi drama po yong yo resta lhopert tanda ane ko chi duidig rambala sakura hachan travi rambala chele kare dawochi yome ko la ya sinyi tu yo rest system balaya ne tanda de la ya ngasuge rigze ta ane ko separate ko lan desija bala tene dete ane neton teta kala ya chikar sugre ta chi tes chi ko to laya girwa ta chi yes.
It's estimated that there are about 20,000 human genes, and a large portion of those are going to be involved in development. So we will not be able to talk about all of them that are playing a role in this, but I do want to highlight a couple of the ones that are most studied and most well known and that we think are, kind of the most important for these processes.
Ta tanda sugi mi gi serpetan drechi kishi shepa imbaina ane serpetan drechi drawa yo chu ane mi la ya dro mi la ya rigze te jeans gro rigze te tinis yo pa ge dus keche shege yo res ro ta te chama nga sugi re ngo ni desha jate yong ma res ta yong ma da te na ne ane khashi chi dus nga sugi mix kar de ta desha jage ins khashi chi ga mix ka ya te yang ko tanda pertanga suge chi takshe simji la chi yo pa ro mangpo chi yo pa te ge shi to de ko to te ya bu yo pa te na singi mi gi serpela ke chembu inju ge ji kha chi du sungo chi gins.
So you definitely will not need to know all of these exactly. I just wanted to provide some specific examples to you so that you have a little bit of an idea of what scientists have been studying.
Ta tanda de desija ba na dita ta kare kare imbate chi shit sopo gane re kare imbate chi chu haku yo ma res wa chi gi chani bana ne rigje de ta kare tan ro yo pa ta de thada ya ane ka chen rupa ge simji kare de chi yo pa la so pa te ngoto chi gis.
And even this list is actually not fully complete. This is already selecting some of the most important. And I have just maybe 2 or 3 on here that I'm going to highlight and give you a little bit more detail.
Ta tanda dila ko pa de na yang ko ka sur ke chembu cha chwa chi yo mares te yang ko dam si chi de ne shita khachi chi gins ta tig na ya khashi chi che sula ya ngoto chi de te khusu ki cheli karim ba te ngoto chi gins.
The first one that I'm going to highlight is the first one that's listed called Hox genes. And it says that they're important for axis formation, but really what that means is it's talking about the body axis. So having all the proper sort of segments of the body. You'll have your head, torso, legs, if you're a human. And so that's what it means by axis formation.
ta tampote kare system bala ya rigzing decision to have the hawks ki rigzi sa te tampote yes the hawks rigzi sa te la ya ngasu ki dus ta ba imbana ko ane jong thu ki trip chala ke chambu chagu yore to se go res ta tanda ngasu mila shisha ba imbana ta jong thu system bala ngasu ki drum shit ta khathula ya thula yo pe jong thu te se go wa ngasu ki go che tongkho che kamba chi te sama ma thula yo wa wa jong thu ki ngo te ki trip chala ke chambu chagu yore
It's very common for this, it's actually a family of genes, but for this, it's very common for this gene to be studied using fruit flies. And since I studied fruit flies when I was working on my PhD, it's one of the ones that really comes to mind when I'm thinking about genes that are important in development.
Ta tanda ke hawks sa te ro hawks rigzi sa te rigzi ki yang dinsen chi res ro family of genes sig dwa rigzi ki dinsen chi chag res ta de ko takshe chi te karila te takshe chi ko system bala nga su ke dambu dwa dambu fruit fly sa gi chi dwa dambu buch dwa o te la ya takshe chi de te la pa ta ngar ngo ma te la takshe chi bi kapse de la ya ta yo rigzi de la chi tag ngone.
And in developmental biology, this is something that would be very common, that people are going to do the experiments using model organisms, such as the fruit fly, or there's some little tiny worms called C. elegans, or using mice, because doing these studies in something as complex as humans is going to be, it would be very difficult, but also then there's a lot of the ethics that come into play as well.
Ta tanda chi dida do ko la ya chitang ge takshe shinji chi wi kapsa nga su ge samchin shenda la takshe chi ge pichum peche tang ge yore sta tanda piche cha pa bana tanda ge fruit fly se do ta ti dambu ti wa dambu chi te o ti ta ani shenda ya ko tinda do peche tang de takshe chi de ta ti ge takshe chi ge pizu ti ne mi gang la sa ri ta ge ne ta te yong ge yore sta nga su mi gang la sa je chi ge chi ma thu pa te yang ko ji kasugureta ji takshe ta yang zangcho tang kuncho shone gege or wata mangpo chi ten resistance tambala ya tanda ge socha jemba gala ya socha jemba ge takshe ge physics peje tang yores.
And then the other gene on here that I'll highlight is this one, and it's called a zinc finger protein. This one is also a gene that's very commonly studied using fruit flies.
Ta shenda chi kare system bala ya rigzi nyawa te ro tanda nibate kare system bala ya zinc finger se do ta tanda ge rigzi de res de ya ko takshayam ko ja chembu chi yo pa kan shila tanda ge ta te dambu de gan la chi yo res.
And this one I wanted to mention just because I think it's interesting from the perspective of how people select the names for the different proteins when they get discovered. And this one is called zinc finger because the actual shape of this protein, it will look a little bit like fingers. And so that's why they chose this name.
Taya tigane ya chikare system ba kutsu takshe chi bi kapsa skorwa ta te tanda de kapsa zinc finger te disease ke she ko wa te ko takshe chi yong bi kapsa ne ko ge min do tan te yan san samala ya ko ge zuye la te ne de ta ke ores ra ne de kapsa ba na ko finger tan zumu sa ta te ko ge zota te chin zumu ko zota te dawo ye cha ne tanda ge de zinc fingers zinc zumu sa de ta ores.
And another one on here that is an example of a protein named for how it looks is the leucine zipper, because in these kinds of proteins, you'll have that leucine amino acid sort of every other, and then it looks like zipper, like if you were to zip up your jacket.
Da yang ko zutala tani de tapate kare system bala ya tiga gang la yo pig leucine zipper la sta leucine sa te yang atsu te nange kasa nucleotide tige rich kirwa tanda kasa amino acid tige rich kirwa kuge imba kansila kuge triptante zipper sakro nga chuge chardok ngo la yo cha ne losing chardok silmindi tu stasha yores zuta la tak yores.
So here I wanted to introduce just a few genes that are really important, and in the next slide I'm going to show us a little bit of a flow chart that will demonstrate how when some different genes are turned on and off, this will lead to the creation of different cell types and therefore different tissue types.
Ta tanda de la nga chuge rizi khachi du sungo to chisha yes ta tige je te la nga chuge rizi data ani ko kar sugre ta rizi te pharju chiwa ta thapon kar sugre thapon chenju chibi kapse ta rizi cheta pa chi 세바다 마세베기 오라테니 있어와 리지 치타바치 세바다 마세베기 오라테니 데테코 타폰카리기 오란 지만지르게 지가 림반도 타치고로 코른도 타테 되게 지라텐기스.
Okay, as we're looking at this chart, we're going to start right up here in the middle center where it says endoderm tube. And can someone remind us what does the endoderm become?
Tanda de kasunga suga nangshun sa sto chimade la tabambana nangshun gu bugula sto ra ta nangshun te karilan jurgre sa digestive tract
Okay, yeah, and is there anything else that?
yenda karilan jurgre sa lungs, liver, pancreas,
Yes.
shema.
Great. Okay, perfect. Thank you. Yes. So there's many different organs that the endoderm could become.
Ta tanda de bana ga suga nangshun te swa kare la ya chenju chi ma chi te la ya ko la ya chigdam ka ma sono mambo do as kurwa.
And so just a reminder as I go through this, I'm not really worried about knowing these exact gene names, right? It's just I'll go through this and it's the idea of the process that it depends on which genes are turned on and off as to what cell it becomes.
Ta tanda de kapsa chesuga rigji chera pa tu minti ngi gugoyo mare swa minte chera tu mangina dikuyo res. Ta chode de la karchi samna tan roche system balaya rigji gi chera pa chi se masala tene dete ko wampo karilan jurman jur sa chigarwa te gi chigin do sete la ya ko alam pachiro chis.
And also, I don't remember if I already said this, but this would be occurring during that process of neurulation. So we're now talking about the third step of the embryonic development.
Tanda de ngasu kawal hede ore tanda deshe jate kawal hede ore system balaya ngasu gi wanting to room says to rimba tangpo de la ngasu gi karsa drimsang ge serpa ge kecheshan stambala rimba sumbach to oti ge sumba del hede ore.
So we've got the endoderm. Differentiation has only just started to happen. And this is one example of one direction this could go. That's if PDX gene is off, that's what the minus sign means, and ALB is on, that's what the plus sign means. The result will be a liver cell or liver tissue.
Tanda de kasura ngasu tanda ge karsa wanten turun ge rimba tangpo te kasimba na ngasu chenjir te ta ko thoma ta nyonyo jonggo sgor wa ta ko thomi chala ya nyonyo jonggo sgor esta de kasimba na ro tanda ngasu ge ta nangshung gi buguch to ga nangshung ge bugo de la ya rigji selon chi ma chi la ten de tan dugo res ta tanda de la ya ko ge rigji te PDXL h to ga one
It's a one.
PDX1 la to that the PDX1 gana ya tra domas doa doma tige ko rigiti seme patengores dosa rigiti mase bache ola ya ALB size doa ti sebares roa rigiti gangeti mase bache o kote sepa imbaina ninga sura kare dukre chimba dukuyores.
And then a different scenario is shown over here. So you can tell in this one, they both have negative signs. So that means in this case, they're both turned off. In that time, it's going to result in forming intestinal cells.
That dela ya tsho sala yesho sala ta ba imbana ko mando re dorwa. Tande ge rigite nisha semi dorwa. Rigite nisha mase ba imbana ngal chagre juma chagyores.
And then on here, this is showing third option is, so it's the reverse of this one, that PDX1 is on, ALB is off, and then that is going to make a pancreas cell.
Tarwa tandage ta ola yopate gangati ge doshore dorwa. Asula ya PDX1 ti ko sebache, ani jenda ALB ti mase pa imbana ne kal chagre shema chagres. Shema ge thapon la change chigres gota said.
Yeah, and so from here, I won't go through every one exactly, but the idea is here that it is getting more and more specialized. So when we have a pancreas, there's multiple different kinds of cells that will even be within a pancreas. So the next step here is that it might be an exocrine cell or an endocrine cell.
다 되게 오라 임바나 가서 셔마 임바나 셔마가 나라야 차포만 보여레스 만다만 보여레스 다텔라 양코 koga nangla ya rigzi chikare yomela sopala tene detis ane ko ya chenju chigarwa thapu mandra chenju chigar tsuju saimba ne endocrine na namen gi thapon ro phajo chimba ne exocrine system bala e chimeng gi thapon chag yores.
And then from there, it can become even more specialized, right? So this is showing you the genes involved in this process of differentiation, that even within the endocrine cells, now there's 4 more options, and those depend on the genes that are shown there.
다 되게 가서임바 다 가르치고 system bala ya ngatsuge nameng gi thapon imba na 되게 나라야 ko rigzi kar yomela tene dete kumana mana. Can I ask you a question? Yeah. Can I ask you a question? You have like here plus and minus, and you don't have plus minus on this.
Oh yeah,
genes.
that's a good question. So these would just be showing that you need these genes to be on, and then it will turn into those.
Okay.
Yes.
That
Thank you for clarifying.
Galaya got so same as it lay cook dogmatong method to go gala talarwa. The all I cook meta dogmatic trigamadoa. The mepatilago yopa Numa but some girls that could Danda Danda Dona de Triala Ichimichi reason data kuyores, reason data yoba and kuchagores. Yes, thank you.
Yeah, thank you for asking. And then just the last one here that I think I'll point out, because it's probably something that we all heard about at some point in time, is that these 4 genes need to be on to produce the beta cells. And this is the cell type that produces insulin. So for some people, if they have diabetes, this is where there might be some problem with these cells producing insulin as their body needs it to.
다 되나는 타바임바나 가서 그 남행 그 타폰라 타바임바나 남행 타폰나 파조 때라야 가서 리지만도 시도와 리지만다와 시티 고요레스 리지만도 시티 요파임바나 가서 그 베터 타폰스도와 베터 타폰도 저고요레스 다 티마나 델라테네 데당 가서 그 인슐린 티체튼 짓고요레스와 다 게티야 델라션 쇼파임바나 가서라야 네 인슐린 티체튼 짓그마트 바이게 네탄라테네 데다 단당 가서라 아니 진유 나사치요로 진사쿠 나사티 용고요레스.
One of the methods of studying some of these genes that are involved in development is basically to either make a mutation or sometimes turn that gene off, and then you just see the result of what happens, because that will give you information about what was that gene doing in the first place.
다 단다 리제 데이터 그 젤리 디라도시 드고 요멜라 소바텔라야 시야테 갈치고레스나 아니 산삼라야 타세치비 값스 단다가 리제 데이터 아니 코라 리제 데이터라야 아니 인지던 거요레스라 다 산삼아 가츠고 그 세기 찍고요 말레스 매프즈고요 레스라도 매그 찍바텔라야 인지던 와테와다 리제라 인지던 데이터 리제 인지던 와텔라테네 데이터 그게 아니 지금 체나마 소나로 그게 남바 간다치 응고두세 트렌디텔라 타세치고요 레스.
So I'm going to share examples of mutations in 2 different kinds of genes, and I wanted to share these because, as you'll see, they end up having a very striking phenotype. So you can really obviously tell the difference that these genes made in the development of the organism.
Tanda dige jela yang suga rigzi la ya ninjir tongwage namba mando anirwa rigzi enila ya ninjir tongjuge pesunching da desejage instant desejage junsen ka rin system bala ya rigzi de la ya jirwa tongwala tene dete ane ko ge ta rigzi ngonam chigorwa tila tene de tang semcheng na tele chechu ngon tang chigorwa chechu ngon tang tela ya ta ko chepra hasan chembu do yores ti instant bala ti dus tengins.
Did we have a question back there before we go? Or no?
좋아요, 회사. 라파창이 서로. Yes.
나 인사 나지 지금 좋아라 on 치고 말해. 근데 근데 off 네가 off 치네 지금 줘야 돼.
Two microphones.
So my question is to differentiate into the intestine cells. We have both the genes off. So usually, in a sense, like when something differentiates into specific cells, we don't need to have the genes on. can we have this sort of like, even if the genes are off, can we have this cell differentiate into intestine cells?
Yes, okay, so this is a very good question for thinking about the process of how all of these genes are working. Do you want to say?
Tanda di bana ro tanda nga su ri de da ke ko ge lekha karan ro chi ji yu me te la ya ne samlu tanga thela ya drop ya buch dus.
There's some, this is a little bit kind of simplifying, but sometimes genes can have two different kinds of functions. Some of them, they'll really be like activators and they're really causing something to happen. And then there's also, actually what I think you'll hear about more on day 6, there's some genes that they're more suppressors. It's like they're preventing something from happening. So there's these two basic kinds of functions.
Ta tanda de la ya chi nga su ri ko la ke je stambala ya ri ge ko la ya chechi number ni ngongu yore swa tate nga su ge je la ya pechi ni ma tupa song song ni ma tupa chi la ya nga ni ma tupa na la ya te deshe ta ju jagres kashi ba na ri yo patila tene de te ko ge ni cheli chi ngon su to po gan ting chi ni tin da la yore san sama ri ge te ge cheli te ka res stambala ya jenda chi ngong man ji ba chi ne ni yore.
Yeah, so it's possible that these genes have that kind of function going on. Another way to think about this is also that when they're both turned off, it's sort of the default that it will become intestines. And then just in order to make it do something different, then one of these genes needs to be turned on. And depending on which one is turned on, it would turn into a different cell type.
That under the desejata chitires rako yan chi karsogre chi sekchibata chi kabala tene da jongo yo parwa chi samna tonta chi kare se stambala ya keti tandage rigzitin cha seme pik kapse nga chu juma chago yorwa te kro tampo kro default se stambala ya ko masage chirtang gu ta chi cherwa ko chirta ta indam bana dina ru chi chago yore swa ta keti is ta rigzin din gi chi ke kano chi se pa imbana ne jenda ngu la jira chi gorwa o thapon jenda ngu la chagu chi gre se te ya kondeshe jenda desejata jemba chi res.
And then just one final thing to note is that within these cells, there will be many other genes expressed. This chart is only focusing on these two, so those two are not on, but there will be a lot of other genes that are on, and so they're playing a role in that as well.
Ya jenda chi kare yore system bala ro ta ta de khola ya ko khase uchi zo pa imbaina tande ke rigzi din cha ko seyo mares ro imbaina ya rigzi jenda mampu chi se de yores. And Tiangu Nigu res Tincha Masepa Jenda Chi Sepal Tende Tayo Kushichin Jongo Yorwa, Tiang Nigu res. What is that?
And that actually is even a perfect connection to exactly the function of this gene, which is called myostatin.
That under the Troatella Tene Tangatsu Jenda Chilatagins, myostatin say Rigidetagins.
And so myostatin is something that actually sort of prevents having too much muscle growth.
It prevents
having too much muscle growth.
Tanda myostatin say exist or the digi karchigre system valaya and atsuga shadrimte shuchambu yobade and a shuchambu mayowachi and gokuyores. Shadrimte mampu pela and gokuyores.
So just so that we know what we're looking at the top, the way that these are labeled is it's meaning what this gene is like on each of the chromosomes. So normally we would think of what is the gene like on the chromosome from the mom and the chromosome from the dad. And so since we see these two pluses here, we know that the myostatin gene is on for both copies of the chromosome in these dogs.
Ko kandado yometi chongores ta tanda de la ya chonta ko phajo salai dom touch don sala dom touch doati kari chongores na nga chu chepon ni cheg gang la ya tanda myostatin se gergidi kanda yome chongores ro ta chepon gi ancha gana system bala chi phani chi mane ro ade ka se tanda de ka sim ba na pha tang ma ni chane yong ju gergidi ni cha seyo parwa seyo pa ba na ane tanda phajo sala yo ne ki gi gang ge namati res.
A little bit of review for you from our genetics classes. What is this called when both copies of the gene are the same?
Tanga suge kasa jingwi chanri ko kola ya shitachi karsa nga su jashu do chi che bai bai na rigzin cha sugro da chromosome gang la yo pi chupong gang la yo pi rigzin cha chi bai bai na tela gar la gres rigzin chi bai na
Homozygous? Maybe.
Reason cha chipana kara phomoni ko mochire.
Homologous mare. Homologous is said.
Tela ka lag raha homologous
Yeah, so this is super close. It's homozygous. So yeah, it's
Homozygous.
Homozygous, yes.
Tela homozygous agres homologous mares.
And then what about, so in this case though, they're different. In this MH, I could have changed, but this is, it's basically the minus, so this means it's not there. So this one has a minus and a plus. What do we call it when they're different?
Ta ta de ka se banar wa ni pa big de la ta ba MH sai te ka se ka da ko na rigiti me pa se ko ra ta salon chi me pa song go res ta rigiti ni cha chi pa me na ka lag res.
Yes. Great.
Rigiti me na lag res.
Yeah, so we've got the homozygous and then we have the heterozygous here. So then you can tell me, right, so just observe these dogs. Which one seems to have the most muscles? Which column?
Ta ta da che su du sta ba im ba na as ra ta ka te a to b to c la ta ba im ba na ding na ni khila ya ni shadu mang shi te ko la dus.
Okay, I think I'm hearing mostly C. Yeah. So I think I would agree, right? C, these are very big dogs. You can see they have a lot of muscles.
Ta da c la re duas ra khi chembo che ani shadu mang duas.
Yeah, and then these, so since these have, they do have one copy of the gene that is turned on, but the other one's turned off. So I think I would agree with you. They seem kind of in the middle, right? These do have more muscles than these dogs, but they don't quite look like those dogs on that side.
Ta kima te la ta ba im ba na kho ge tan da shadum tan ro pe resist chi se yo re jende te se yo marwa te la ten de te ani khulo dus ta ba im ba na tan da katki tampo na yo pa te le katki nyo na yo pa big na yo pa te la ya shadum ma do ne big na yo pa te le ani sig na yo pa te la shadum ma do as.
This has been observed in other kinds of organisms as well. One of the first ones, at least that I know of, was actually that was observed in cattle. So here you can see a cow that has this gene mutation and that it has developed lots and lots of really big muscles.
Ta rizi de la ten de te shadum gi kor te la ya ne social jenda ga na la yo res ta jenda ga na la ya tampo thoya te wa ani ta da de la ya pa chi to wa balance to ga de na thon yo res ta ta da che ba ba ne zubu kanza ta da shadum re do wa.
And as I mentioned, a lot of these things involved in development, scientists study them in model organisms. So that's what this is showing here. You can see, I think pretty clearly, the difference between these two mice. You can tell which one has more muscles.
Ta chari pa zo ge ta da du te la ya ko to ya pu yo ga che te ani so cha ke pezu tin de peche ta da ta se chi ge yo wa ta ta se chi ba te de ta da de tsi gang la ta se de ba de ta da de tsi ni cha ta ba im ba na gang ge chi la ya sha dun chi wa se ko ta ma yo pa de du ngo so to bu thont kan du wa sin de ga ta na.
And then just the second kind of example to share, this is now, we're coming back to that Hox gene that I mentioned before, and that I said they often study these genes in fruit flies.
Ta da de wa shenda te rigzi shenda te ka na Hox rigzi sa de res ta Hox rigzi de ta che ta da de ba na ga su dambo gong la ya ta se chi yo res.
In this row, so this is actually, it's the same picture that we have up here at the top, but there's one that's in black and white and one that's colorized. And since we were talking about visuals, I thought that I would include both, because to me, it's a little easier to see the differences in the color photo. And I wanted to show you the difference.
Da da de la ya ga su ge ko ge parti ro pha jo la yo pa parton se jo la yo pa ge parti ya chi pa yin ro ani pha jo la yo pa te chontra me pa che se jo la yo pa te chontra yo pa te yin sta de ko sha go ge jun sen de ka rin se sta ba la ya re ga su tang ge se go ro ta ga su ge 지금 통 태하게 비선 배치 땅 바에나 리라우요를 세시거와 되게 나네다 나라 천 바에나 네 천차 요주디나 다 바에나 아니 쳇버티 통야를 리라우두스
So on this side, we see this is what the normal fruit fly would look like. So it would just have some antenna right here on their head.
아하 다단다 파조사게티 카레레스 담발라야 아니 남진게 가속에 따 정보그르기레스와 타고 고강라야 가속에 초라스그로 또 그 초야가 라치로 싶다 어때 남진 요바된 고강라 주도스요레스
So as I mentioned before, these Hox genes, they're important for that body axis formation, so kind of knowing what goes where. So in this fly, when it got mutated, now the legs are growing out where normally there'd be antenna.
다 단다게 디스그로 다 액세스 시스템 발라야 가서 jon트게 단다게 Hox게 리제데 가서 jon트게 트차탄도 시스템 발라야 티카스그로스나 jon트게 나네 다 고름 티쿠게 땡케기요 레스트라 다 단단데라야 수족게 데라타 바임바이나 아니 코다 단다게 Hox 리제데라야 지와탄 요자 아니 고기 남제 라티치사게 고기 차시텔라야 가르치다요레 깜파치다요레스
So this would be a case where before doing the experiment, scientists wouldn't have known what was going to happen, but they ended up making a mutation in this gene, and then they saw that the flies turned out like this, and so then that's what gets them thinking of like, oh, these genes must be involved with something that's doing the proper order of the body formation.
나하 타코 담보 탁세 담보 집이 값심바나 데 영아 싱기요 마레스 차르파기로와 임바나 다 탁세 지지 데데 리그지텔라야 아니 저와탕 데테 아니 단다게 담보데 디라도 지존 스탐발라야 알레타 리그지데테 스탕아수가 즈푸게 존트케 탑차간라 타바 임바나 아니 지 연락게 파는 릭탕테탄 데와도 세데 쿠스 싱기요레스
So there's many different kinds of Hox genes. So this is a mutation in one of them. This is a mutation in a different one. What do you observe that's different about these two flies at the bottom?
아하 다단다 이거 Hox 리그지세테야 코 리그지 만도 망부요레스다 코 담보크로 딘센치라 토고도 알아 다 담보테 치기체 단다 오텔라 타파임바나요 Hox 리그지나네 만도 치기라야 니저 담바레스 다 오케 पर्टिंशल ताबा एम्बाना खेबर करे दोसा शोभा मैसेज एंदा
Oh, this is the wings?
aha
Yeah. So two sets of wings?
yes the what the feathers what do you call this one
Yeah, just like the wings.
yeah the wings yes they has 4 and the other one has just only 2
Yes, yeah, exactly. And so you've exactly noted about the wings, and I'll even point out, it's even this whole thorax body segment. It has 2 of them. So this whole middle body segment, it just got duplicated. Instead of making only 1 like it normally should, it made 2 of them.
right that is shoppa manda shoppa tambutela ni che sujotela ya shidwa te nanshinge ne ko shoppa thosa ge zupu ge chashi te imbaina ni do andela ro shoppa thosa ge deshi tan deshi do ha
This,
so like
this little
small-
one, the
yeah.
haltier?
yes
Yeah. So that's a very good observation, and so this is something that's kind of unique to fruit flies, and so this here it would be this would be considered normal development in this case, but yes, exactly, it's showing that instead of staying like this, which would be normal development, okay, now it actually that ended up growing into the whole other body segment.
this one is the mutated right
This one's the mutated, yes.
okay ta chitangi baranga sugi dambu kola kicha shan stambala ya phajo te ko shobba ko ola kampano chinchin da lapando chanchon yin doa din ba na phajo sagete ko ta dambu no ne chirtan khorang ga chalon chi tan ba na zu pu tja din ro chi yore sra ta tanda tjo de ba na ko ta ninjur te tjo de do ka istabale din la yin jir de yore standa
So for the scientists who were studying these, they keep going through and they're making mutations in these genes, and they see, okay, the mutation in this Hox gene now makes the legs grow out of the antenna, mutation in the different Hox gene now doubled the body segments. As they keep going on and doing this with all of these different genes, that's when now they're collecting all this evidence to finally say, oh yes, look, these genes must be involved in development and the order of the body.
Aha, ta tanda tsaripa chu kutsu takshi chayati tanda namba dina gani chigu yore srwata ko Hawks rizigna ne richigla ya ninju tangpa imbai na ne ko ge chorra chisala ya kampa chigi du sayatang roa ne keti yang shenda chila chepa imbai na ne zupo ge kasa ta ko shokpa chisa ge tripchati kuro nisa yongu du dab ni yongu du sayatinda ro ne ti nanshin ge de tanda ta Hawks rizig di shenda la ko takshi chigi ane ta ko nyamtendo chigi diruchiyon stambala ya Ota tanda ge ta Hawks rizig la khonsu topa ge data thamala ninju tangpa imbai na nga tsu ge zupu ko jontka gane ane yenlak ke nesa te tagla ya sukor ata yenlak ko kurum te tagla tang dewa resha te khud la ninju tangpa imbai na te tagla chun chig du sa te kusu takshi chi du snye ki yores.
As we know, it's always a combination of genes and the environment that are going to be affecting organisms. So in the next slide, I have an example of an environmental factor that will affect development. And this is a little bit related to like your question earlier about how exactly are these some of the signals initiated.
Ta tanda ge dela imba na nga su ge rizig ge korla shek yores rizig latene dete ane ku ge chigi yanlak triptang la soba de desija gores yang shenda yang ko yanlak triptang te kola yu khoi kishi chen chaya tina do yang yong yores ta tanda de ge jela yang asu karita ge insena ane karsa tangpo te la ya ne thapon te ko chenjur doya diskora thapon chenjur doya la ya ane dati ko ana yong gores stambala dati khoi ni yong gores tangpo trach tang song as khoi ni yong separe se te kola yang asu ta ge ins Gela has a question.
Tanda di dangma shobani cheba tu pike di raju cheba sta. Di taje kono sonjoro es jin jowatane.
Are these mutations created in the lab?
Yes, the pictures that we're looking at here, this is all created in the lab.
Taje kana zojo res tanda prayochi de sama. Do we have the Hawks genes in a human?
Thank you, and I meant to say that also. Yeah, definitely. Humans, we all have Hawks genes too, and they can also sometimes be mutated. In humans, one of the most common effects of a mutation in Hawks genes, it's not always as drastic as this, but it would be like fingers get stuck together. That's one of the effects of a Hawks gene mutation in humans.
Try abudus tanda Hawks rizite mignala yores na yores ratanda ngasu mignana yang kashi chiga Hawks rizite la ya ninjur thechu ge netan tenda do yong yores ta ti gi ninjur thechu ge ta chi ngonam ti tenda do ngonsu to po sugro ta chi ngonsu to po khetsu tenda do ta yang ngonsu to po tenda yo mares imana yang de la ya Hawks rizite la ya ninjur chong la tenda de te ngasu mignane ka sansaman zumun jer de ni tenda yong do wa o tenda do yong yores.
And I thought of one more response actually for Gailey's question. The cow that had all the muscles though, that one did happen just like naturally, that someone ended up observing this cow that had so many muscles, and so then they wanted to figure out what happened to cause that.
Da ya tanda karsa de zuare se te tang drechi de tang tangi dina la ya te palanch ten so ro langos ten so langola ya ko ta ka shadmish ki chambo yobate ne zuju tina mares ko ranjang ki tina yong de ta ne megi ale de ranjang ki dira yong wate karila te ne inas le takshe chi de tu sa ko jures.
Okay, here I'm actually going to ask you to tell me a little bit of what we're looking at here. So what do we see on this axis? What do we see on the y-axis here?
Ta tanda de la remochi es ro ta remon de gane chechu ge kare thong dos te ga jonthi gala kare dosa jonthi gala kare tsha sa kanja manja like that. Yeah, proportion male.
Yes, exactly. So when we're looking this way on the y-axis, this is going to tell us what proportion of each of these animals are male. And here when it says 1, it's really mean like 100%. So up here means all male, and down here would mean all female.
That under the positive karsa proportion male said the Pugi Durses Gores Ra Pugi Durses at take it to Shigores, that under the Kose and Bana Yara Gangan de la Chichdoa. Chitila Lesona Karsagaya, Gangan de Lesona, the Jontugan Segorda, Jontugan Gangan, Chiosati Kapsa, Kanza Pure Segores, Tinajinke Jontugatama Wall, Lekoyo Setilaya, Kanza Mure Segores.
Okay, and then how about the x-axis? It's the same for all of them. What are we looking at there?
Hanta de Gas de Tetika Gala Kalak Dusa.
Yeah, exactly. So this is a really interesting phenomenon that happens with reptiles, is that depending on what temperature, in this case the eggs are, it will affect the proportion of those offspring that are male or female.
That under the Casa Tetka Galaya, Trosetangdua. That the Casagore Senaroa, that Kogi Dana Tundi Socho Kuzugi Gunga Shasachuaroa. Gunga Tansa Tigi, Gunga Shasa Tigi, Troselatanidatis, Roa, that Troseti Tisagora, the Troseti, Tilatanidate, and a Kota Gunga Keli, Tundi Socha, Chivi Kapsa, and a Pull and Jugre, Mulan Jugre Sach, Goroa, what the Tanguris.
So I'm going to ask you in your groups, talk with your team, and for each one of these, A, B, and C, we'll write the temperature for where there would be the most male offspring.
Hanta Danda de Casoroa, Chesu Caretaro Sena, and a period Tampoche, Nuache, H, B, C, Tinalaya, and a Casa de Pum Manshu Pigi, Troseti Carin de Taroches.
And since we don't have the degrees on here, you can just write it as low, medium, or high.
That under the Galaya Troses and my sopi, Trosetilaya Suki Troseki, Andratus Tremendo Astra. That is Tambalaya Chisu Karichin and Grace and Apa Dustabano Yonshosani Roa. Any Trosa Mawache, Drenwache, Trosi Tuas Lieutenant Gores.
Yes, go ahead, take a few minutes.
That read the Namaches. Yes. This is Rocha. That is Rocha data Dada. Chesula Trotica Sekacha Mesro, which do Usra. Chesula Roa. Tanda Trotica Sanarwa. Chesugi de la A, B, C, Nani, Roa. A, B, C, Nani, Kangi Manshu inside Tesago to the Chik de Setimares Roa. Ela Imbaina. Trose Tuaredo, Mawaredo, Casa, Drama, Casa Pumando, Titares, Bilaya, Bila Chipula Tares, Ra, Pumate, Trose Tuaredo, and Dramaredo, Mawaredo. Tinajiki Selaya, Tuaredo, and Dramaredo, Mawaredo, Susu Susura Tagores, Suncha Drinan Chigita Mares. A good ada? Oh, pay up for the touch with the Chistat.
Thank you for all of your good discussions of these graphs. We're going to come back together so that we can review them and hear what your thought process was.
That triple pay apunasos, that's some yar zero chase, and it's a correct to the chiomete yamtichtagosa.
Okay, the question that we asked is, at what temperature are we going to have the most males?
That Trotica system balaya, Trosekangi Kapsus, Rada, and a poor Manshiores, Troteres.
So we're going to look at each of these separately. So these are 3 different graphs for 3 different organisms.
That Andangasuke, 3 Laya, relation to Tagins, 3 social manamana Chongdua.
So as we go through these, you can just shout it out, say, and it'll be low, or intimate, but low, medium, high. So we'll just shout out what we think the temperature is for most males.
Poor Manshi, Yoyati, Kapsanga Sugar, understand by Bina, Nichesuki, Troseti, Mao, Jingwa, Toes, Leniniam to Lavrochi.
Okay, for graph A, at what temperature is most males?
Low.
I think low. Yes, yeah, exactly. So here we have low temperature, and then we can see on the y-axis, though, it's where we're going to have all males.
That Amputela in Bana, Trosa Mau in Bicapsres, trotse mau imbe kapse ane tanda trotse mau cherwa tanda trotse mau te kange kandare nga su theti kanlarwa theti kanne trotse mau imbe kapse jonthila ta ba imba na yara chis lagdo de kapse ta chama pu imba teres.
And then just for this one, I'll go through each of them and then we'll get the answers for the others. But if we move over here to medium temperature, we see that we can go up, and on the y-axis, we see it's going to be half and half, males and females.
Ta tanda nga su phajo chagirwa thire etila ta ba imba na ane nga su trotse dringwa yo pikapse sra trotse dringwa yo pikapse ta ba imba na ko cheka re do asta cheka re sena karse gore sena cheka potna cheka mores.
And then if we go all the way over here to the high temperatures, we see that the proportion male is all the way at the bottom. So that means these will be all female.
Ta tanda trotse ha chang thumbula le pikapse sra trotse thuila nikala le pikapse kare do nga chu ta phogi durche tande bi thirte ma wala le do arwa te le stamba karse gore se stamba la ya trotse thumbula pikapse chama mu imba tengyores.
So for graph B, what temperature has the most males?
Ta thire bela ta ba imba na trotse kare imbi kapse ane pumando es high. Yeah, high.
Yeah. So the high. So at the hot temperatures over there, we go up and see the highest proportion male.
Ta tere duas de ra trotse te thur do ba na trotse thubu yo pikapse thua yo pikapse ane pumando as.
Then graph C looks a little bit different. So what temperature here?
Ta de thur sidi mando re shas ta de kapse ane ko pu ma yo pi trotse te kangere dos. Yeah.
Yeah. So it's in the middle of this one, right? And then we're seeing that's male. And so if it's really cold or it's really hot, then we're seeing that the crocodiles will be female.
Ta tanda de kapse me trotse jong yo pikase pu ma che ane trotse chiga ma wa tan trotse thua yo pikase moti ma te ngudus.
All right, so we're just now finishing up these, but we got through talking about the visual representations and then also those different stages of embryonic development. In the early morning session, we talked about cleavage and gastrulation, and then those processes that we just finished talking about would be mostly involved in the nervulation.
Ta tanda dina ne nga su ge mugila taba imba na tampote la ya nga su ge thong rong ya ge pisun peche tang de tan zanta thate kola deshi je yore tin ang sing ge nga su dum sang ge charpe kaps ani samjir ta tanda trodun dermenti nga su ge tampo deshi je tanda imba na nga su ge dikarsa wanten trodum chigorwa o te kola deshi je yes.
And oh yeah, go ahead.
tanda dis da ge da te ge dinalo Rubedo solo popo machaya de ko sarangi shukitik da popo machaya ge rizil loga yore yamare stambu thadi.
Are they so in the example you have for the turtle that in the example you have mentioned that their gender depend on the temperature. So do they have a different genes?
Oh yeah.
Male or female?
So here it will be a lot of a difference in the expression of the genes. And so it ends up being pretty much like some complex hormonal signaling that is interesting. We won't go into all the details, but yeah, it's mainly about the expression of the genes. So it depends on which ones are turned on and off at different temperatures.
Ani rizi kangi chise masela ragli gyo resta ti kapsim bana ko tosela tene deta nga suge hormone system balaya menji kare kare mong chigalo malorwa system balaya ko ge chi jetanta nyong chau yo resta so de ta bana ko ge menji la tene deta kare chi selon chi machirwa rizi na kangi chi selon chi machila tene deta ta ko pulan jwa to molan jwa de yongores
Manzo mila x to y ji loga yara ji tinoji kol yare.
We for humans we have X and Y genes. Do they have X and Y?
Yeah, so they would not have those kinds of chromosomes like that. Yeah, humans have X and Y chromosomes, but for these kinds of organisms, yeah, it would be determined by the temperature.
ta ko nga su mi la ya ta chue phon ta x tong y ta yor wa nga su ro mi la cham ba na tan da ge tun de so chu ko su la ba na ani yo ma res ro ta ko su ko pho la jur mo la jur sa de ani to sa la ten de ta chapter zu yo res
shopa tanda maso stem cell level ni kable ya ta chik so chashi kar chama cha se te
aha
chi dala danchen da de an chi dala tu ban chi ke so
aha
te nan de ni des tran song yo an do shi to ta ko chu ba ge ten da ge da ma re se chi des chi ko da wi cha yo re yo ma re
ta tang ge signal te na lo signal la ko go
na lo ki sin
aha
ina ya ta na lo ya
ta kare da so yo ma signal tang
ta
tang
che
da
sang ko kar cha ma cha se te ki chi kar sa ta jen do shi ba sho ge ne ga da dan chen la ra le ya chi cha
aha
do
re re
te chi ban chi cha wa na se mo chong sai ma ri yan
da te chu go ra ta tang bo de la nga su ke system ba la da te na lo ka da la se ko ko lang go ro ta tan da de la ya chlo ko da chi ro ta de
de on de se tanda di tanda ta da se di chlo da se na ko kar se ta ko nang ge cha se se on de cha se de chno ni ga le yong chu ra yo de
aha
ina ta di kab le chi ge ma ani chlo le ben re ben de ma tang se wi ji le
aha ta
gonga tang se wi ji le cha se de ge wa tang ge yo re be yang na mo ni yo re yang na di ti na ru tu gonga tang se wi ji le cha se temperature cha se garam ba wang ge chi ge de po mo je wa la cha bar tang ge yo re yang na ma chi ko rang ko ba na yo bi ka
aha that te ta da de tru go tang bo de la yang ko ka sa ko da chen tin jo tang ta da chen ge chepra ko ne tru go re te chi par se go re tang bo chorang ge tru ta de la chila son tang san go ra
de nge sam lo kar ko re se na shoba de ma kang cha bong ne ba de kar kar cha ma cha se de dan chen ji le de ke shi do ta ji da lo da kar ina ta din da wo de shi nge thong ra tan de yang chi
ale de ta da chen chi lo ko
dan chen ji le
okay so it is like two subcategories in the question. The first one is you have mentioned in the one the cells are in the stem cell stage. It has the potential to differentiate all sort of cells, but it will differentiate due to the signals. So and in this sense, can we consider the temperature as a signal for the reptiles?
Yeah, I would definitely say that we can think that the temperature is a signal because the cells are sensing that temperature, but then they're creating a lot of chemical and protein signals, hormone signaling from there, yeah.
That ko la na digdu sarwa ta de ko se ba na ta ko ge thapong ge to se de chogo to se de tem ba na ten de ta nga su ge na ko ge ji ge da tang zu men ji ge da la pas mo mong pu chi tang ge res ra. That's the first question. Then second question is like when you talk about the temperature when the eggs are already laid out or even before the laying out. When you talk about the temperature for the hatching.
Yeah, I think they've already been laid, right? The eggs have already, yes, the eggs have already been laid.
Ta tanda de ge trose ge yang ko ge chapar zu ge sa te gunga tang no ge jela res gunga tang no jela ta gunga te tru ge su go yor wa o te ge ma te la se go res ta trose de
ge do to chi tanda di jangma ge ngo ko chi do ga jangma ge ngo ko chi de exo os ko no ri de yalla ga tang zhen ko ga tang ngo chi de chi ge jo re ngi mi ge ngo chi sha ga da ri de jab di gang la jangma ge ngo company to do ga 또 감바 토야데 다 당전지야데 추조고 제레 고체데 특디가랑 고 체트 조조레다 고 클라마 쳔고 치조가 땡 이미지 지조레 가르 감바니 토야데 고라 토도가
가라치고 차 감바 토야데
땡 이미지 제레 고 체트 고라 치기가르다
so 알레다티 드와테 코르가 taksege름 바텔라 타고와 바하 kuchirwe ane hama ko koranta So, for the fruit fly, you have legs instead of the antenna. In that example, is that intentional experiment, or that is just random outcome that because of a mutation?
Yes, okay, so that is the result of a laboratory mutation. And I would add, though, I think the very first time when scientists made that mutation, they didn't know that was going to be the result. So they made the mutation just to test it, and then that ended up being the result. So they didn't just think, oh, let me get legs up here where the antenna should be. It just so happened, they tested the mutation, and then that's what happened.
Also, they know the genes are related to the axle, the order of the part of
Oh,
the
so
body.
they wouldn't have even known the genes were related. For a lot of times in fruit flies, they'll mutate some genes and then they see what happens.
Okay. That the ngimeres that tampokro allergy data kuge azupongge ngatsuge yenlak pangrim tang dewa yore tin stamba data na kayo te sagi omares ta kachirtang imbaina ngatsuge dambo gola ya taksha chete man shishu kusuge tampoko allergy data dewa yopala yang shigi omares ta getting a reason dela ninja tong baina dele chepar kari jug du se to tus taksha chigi yores ta ko ngime do res tampo digin di chongres de chiju mares ngime res man shishu ngime gane chigi yores.
Okay, I think one more question.
디겔라 안저 디텔라 퇴두시라 퇴와지더라. 단다 리베야길라 리베디 공아디 사자 장사라 땅we냐니 참마 그래 디기라 포기기더라. 다 계시 사자 차워에서 지라니 리베디들라니 가서 포제요 말아 다 포차회기다 니와시라 미디갑슬라 손도 리기 데니 마긴딩 하네 차워레스.
So it's like not only the hatching of the turtles, but in an environment it has a really hot temperature. It's a high temperature. In this case, we see the high temperature. We have only females. So how they will have offsprings to have that inheritance?
Yeah, I think this is exactly getting at what one of the concerns is about if the global temperatures are rising, that, yeah, exactly, it will keep getting imbalanced in those cases, and so it will be more difficult to have a balanced reproduction. So, yeah, that's exactly one of the concerns that people have.
That Tanaja restaurant, did and that Serupagana Kolaya, Semtre Yonagan, Etachi to Chagu restaurant, that you Tochella in Jurashi Chambu Tele, Hachan Tau Chabata, Hachan Tamu Chapa in Baina, that Tege Koge, Pomoge, Dorsetela Chepeski Chambu Zugudwa, this Tambalaya, that Koge Riginte, that you Junichi Alaya, and then Getchin Zugu Yosante Telaya, and Semte Chigi res. Sente Chisaga Nichi Chagu rest under there.
지당디 난소 남배 슝기 따술다고 제와이나 드게를 깨지 시시고와 난소기 지당 깨술들다 용라기 전에 시시고와 날게 공게 주게 드게 신이 시시고와 지당 드레를 깨디 난소 슝네 직시에도 시기yor다 다 이나양 미당디 쪼쪼디 당 난소 쪼 날게랑 샤기라 마도 드게를 슈구슈구 샤도고 요마레
미다 자고 말 데스마 톤데 소처도 고아기 친네데 이야데데 공치 우라 두고다 데조 임바이나
디조 공게로와 다 단도 고소기다 드레 체중이 개발지 시기도 와
레레레
베나 디지 슈와이더라 베는 제디치기 공학계로와 아니 제디치기 드레 가야 마다 와체 자 디조기 전에 고 가서 공활래 고 제디기야 되네 용쓰기로와 드레메 돈이 디니 깨디 연구연하면은
드레메 바지 자티가 공안하네
제디기 공화디게 와 제디 고랑기 될 드레 맞대 와체 아니 그 공화도 샤샤와 있는 될 제디기 이렇게 말했어 줘라
so my question is in the Buddhist philosophy, when we talk about the birth, we have like 4 different types of birth. One is like through eggs, the hatching from eggs, and the other one is one of those is the birth dependent on temperature. So it's pretty much similar when we talk about the temperature dependent, the environment dependent in this case. And so my question is, if a bird or if a hen lays an egg, and if the mother doesn't give feed to the egg, so will that egg hatch to a birdie or a small bird? Will that egg ever hatch?
Is it so this like the same species?
Yeah, same species. So this is, yes.
So it's just saying if there's
So
not
a mother bird lays an egg, and if the mother bird just laid over there, and it didn't give any kind of temperature heat, so will that egg ever hatch?
I see. Okay, so I think most of the time in these cases that probably if this egg were to be just totally left alone and not be having any heat, a lot of those, they won't really survive. They still need to have some of that care. They need some of the heat. So there is at some point it's just too low that it won't survive at all.
The survive meaning the egg?
Yeah, like the egg, yeah, it won't survive, so the egg wouldn't even hatch if it's just left alone and the mother doesn't provide some heat.
Ta tu shasha pai bana ta gwate ke li chatu chetina yong maris taman shim bana gwate tagana ja ko ta ko chuni ko chit kamaris ta chitang ke chuni chia la ya ta phama ko gwa rimbala sopa ne ta te chungu ge ores chamuge.
Okay, Landi.
Do you want to go forward or do you want to discuss? Because we don't have much time.
Yeah, I think we'll take one more question and then I'll just go through the rest a little bit quickly.
Trochi language ta tijla method rugies.
당연히 trochi tora. 어디 레벨 도동데 전에 자와 공군이 진 당다디 보도 보모 모유 제소. 대노신 로물에 찍버려야세. 아주 딱 노춘을 가르려고 그래서 나 까바 동보 가서
야 당야마 당봉 아주 참마고 초콩 요슨 진짜 차.
노춘을 가르려고 그래서 나 까바 동보 가서 무슨 지기 일로. 디디포마도 보모 메려 버다. 노춘을. 아니 치스 갈래갈래 아니 다 잡아서 딥종니 아니 보모 종조래. 대동데 이티는 저 둘랭이.
지치 세나 가서 그래.
동보 지기です.
지치 세나 가서 그래.
지기 세나 다지.
This is tanda dekas magic right?
로마다로 동부 지기 차고래 아니 디디 뿜마도 메서 라도 다 사이나도 포인 비차로 그러다
포요세마다 갑디디 아니 포모에 남사요 말해 지체래 거와 동부 가래 지체래 세마다 포다 모에 남사데 같이요 말해 포인세 같이요
저 가르래 도 그래 근데 지시면은 하루와 정말 지시는 하인 버차거라. 지시 정말 하루에. 다 지시 세대 지세대 진출라 고거라. 지세는 그래 진출라. So over here for this reptiles, the gender depend on the temperature. So his question is, for humans, does this apply? And he says, like in the Buddhist philosophy, when the first time the humans formed, we call that instantaneous birth. That human body is instantaneously fully formed. So there's the other sort of understanding or there's philosophy about that. So his question is like, can humans, the human genders depend on the temperature?
I see. Okay, yeah. So, I mean, I think this is a great way to make some connections with what you've learned in Buddhist philosophy and also what we're talking about here. I think from the studies that I know of...
Tanya, listen, please.
The studies that I know of that have been conducted in humans is that we typically just tend to think about it that it's purely going to be the random chance of the assortment of the father providing either an X or a Y chromosome. Up to now, at least, I don't know of any studies that have found any sort of influence of the temperature. And partly, or maybe even mostly, I would say, the big difference between these is that in humans, this is all happening inside of the mother's body. So we've talked about homeostasis. It's a very regulated environment, very protected environment. Different from what happens to these eggs. They've already been laid, and so they're sitting out, and the environmental temperature will affect them more.
ta da che su te draw ta gompa je ta te ya go res ra ta da nga su sen ri na na jang ka ri jang ba te ta ne na ya na shwe na na do chu pa tu pa la draw ta ka do as che gi te ya go res ta da chitang ge ta kar sa gut se de la shisha pa ba na sen ri kin do su la shisha pa ba na ane ta da kar sa go re ta nga su ge draw mi chi ko phul an jur ta mol an jur ti karila ra le gre se stam bala ya kham kapru ro phali chong wi kham kapu ti na le ya ane nga su chu phon ti x te ge do y te ge do te la ya ra le ge yo res ro ta ti me se shen da chi ba na ya ne tang ko lo ga res ta nga chu ba na chi phol la jur ta mol an jur se te ya ami gi nge nang na yo pick up san jong go yo res ami gi nge nang na yo jong bi kap sin ba na ami gi nge nang ge khori ti ba na ko tangzin shu ya pu chi sha ju chi res tangyong gu ngu la ni go yo pa chi res this tam bala ya te kap san jong go res tan de ta la im ba na tin do go tangyom tangzin la so pa yo ma res khori ti ro ko ge ta ka ma ko mo te chlo la tang che ne chlo re ge khori la tin de ta ko su ta chi go yo ro kong chi go ro te res.
Yeah, thank you for all of these questions. I'll do an introduction to the connections between cells and tissues, and then I just want to then make sure that we have time to get to lunch. So we'll just talk a little bit about the cells and the tissues.
Ta danda the nyongkong ge shela parsa ma ta ngol la ya thapon tong phon tren cha ge dreba ko la ya tis chung to chigins.
So this we've sort of already mentioned, but just as a reminder for what is a stem cell, we have just that single cell, and we saw these in the early stages of development that during some of the time it's replicating itself. So this is what we saw during cleavage, but then later on it will end up differentiating into many different cell types.
Ta de ka sang asu tang po ta wa na shen ro de ka sang asu thapon ge thapon chi ne ta tang po de ba na thapon chi de thapon cha gu chi ka se simjir ge kap se ba na thapon su ro chi ge yo ro as ta tin na shen ge yang shen da ka ri yo de system ba la ya ne thapon te change jo la ten de ta thapon kan chi de thapon ge ma da la dus change chi ge yo res.
This is a reminder that I think we have talked about before, but this is a really important point and also very interesting scientifically, and that is that all the cells in our whole body, we do have the same DNA in the nucleus, but as we know, of course, we have different cell types.
Tanda ke ne tonde ka la na cher kana ne ko yan chembu ge ni chinga cha go ro ke chembu ge ni chinga cha go res ta de ba na nga su ge pu na na yo pe ge thapon changma ya ko ge dining ge na la ya DNA chi pa chi chan yo res yin ba na thapon ku chu ngu ma da cha go yo res.
So I'm going to ask you, how can this be possible? All our cells have the same DNA. How are we getting these different cell types?
Ta che su la dramatic in system ba la ya ane nga su thapon changma la DNA chi pa yo res yin ba na thapon ma da chi cha go yo ras te kan da su jong go res. Gene expression.
Yeah, exactly. So it's the expression. This is what we talked about with where the genes can be turned on and off. That's how we get all these different cell types.
Ta rizgi ge ko ngo ma ngo ke che shegi res ta ti ba na tang ge shepa na je khashi chi la shepa na je ro rizgi khashi chi se ta se ba ta ma se pe ge number la te de ta thapo ma ndo la juan de res.
So we've been talking a lot about cells, but when you have many different kinds of many different cells of the same type, those are going to form a tissue.
Ta danda thapon ko la ke che res thapon ri chi pa te na ma bu chin dio la te de ta nga chula phon trip cha go res.
So of course, we as animals, we are multicellular, and our bodies have all of these different types of cells. And this is again related to that structure and function, that our cells are going to all end up having little bit differences in their structure, and that's going to be well suited to whatever function they need to carry out.
다 가서 미 바이바나 타포 몬당 소치 차고라 타포요 망보요 레스타 타포 망보요 바만데 디캅스 다 타포라 템비게 가서 그디 기체시그레스 네 차고당 셀리게 대양 만다 용고 레스.
And then just the second point is reminding us that these cells are going to be functioning as a unit. So they're going to have to carry out a task that an individual cell couldn't do on its own. It needs to come all together to form the tissue to carry out the function.
that nibade kare system balaya phone ta thapon de ta nyam tu chho de ta phone tre gung ola ya khosuge nyam tu chho bala tere de te cheli chik duk yores.
There are 4 basic tissue types, and especially since we have lunch soon, I'm not going to talk about these in detail. However, tomorrow, Miguel will lead you through a lesson where you will learn about neurons much more in depth.
ta da de la ya nga so ge phone tre la ya mandwa rig mandwa shi yores ta de ta ju shi mes ta sanye bane ge migola ge chechula wangsa ge kolan de ja bi kapse ane wangsa ge phone ti de ja gres.
And for now, I'm just going to mention and then only share one example of these other 3 types that we have, which are muscle tissue, connective tissue, and epithelial tissue.
Tanda chala ya nga su ko phone tre ge jenda simti ko kola ya ane ko pichun chi re chi de ta ngutun do de chi yes.
So for these, if you're curious, you do have some slides with general information about all of these. For the sake of time, I am only going to talk about muscle because there's one particular slide in there that has a little bit more complex information that I want us to talk about together. For the other ones, I think if you're curious, you can just go ahead and take a look at them and let us know if you have questions.
Ta phone tre tanda cheda tita kola ya dus dema dina do nyenre dida tatu chi sai es ro ta tigna ne kya ya che chuge jela ya dus ta chi china de res ta tigna ne cheto pa tanda nga chuge shadring go kontrate kola ya ngutu chi chi gi ins ta tigna la dema gi na ya ko shadrim tan drevi gi ko ge lekha ge thene ya tis gunyok pu gi ne ta chi us de de ja ins.
For muscles, sometimes the most common type of muscle that we'll think about would be sort of what we were looking at with that myostatin mutation of like all of these muscles on the outside, right? That we can contract them, we can move them, they're what give us our strength.
Tanga chuge shadring ko phone tre kecha shepe kapse chitang ge bana ta chi cha bas ko ra ta yondra ko ngo gane de se jabe kapse nga chu la pa ge na lo la yo pa ra ta ko changkin chi thu ba la pang ku chu tang thya ta de yo chu ge shadring ti ko res.
Those kinds of muscles are skeletal muscles, and they're shown in the middle.
Ta tanda ti ka res system bala nga chu ge ranger ge shane res ro ti tanda de la ten sha yo sir pa tan jer de nu ge shadim ti res.
Sometimes a little bit less commonly known are these 2 other types of muscle, and that's the cardiac muscle tissue. So of course we need some muscle here to keep our hearts beating, and then also smooth muscle tissue, and as shown there, that will be involved in the intestines because we need muscle contractions there as well.
Ta tanda nga chu ge chitang ge shadrum ki chang sta bala nga chu ge ranger ge shadrum te ko che se go res ro shadrum ki phont ge na ne ya senda ni res chi kar sen nga chu nyang ge shadrum ge phont te res ro te nga chu chitang ge ta nga chu nyang ge lekha cha la ya nya phardon che te chawa chi go res senda chi kar res system jamsha sa te res ta jamsha sa te nga chu na lo la chi wampo ga la yo pa te ane ko rangu gi ngucho ta chi goro nga chu ge hama ko nga chu thapchi mata na ko ranjing ge gude chi goro tanda de la ya wame gi tang se ge do rangu gi chi go nu ge dita ta de yo chi ge shadum res jamsha te.
Since we were talking about the genetics that are involved in the different kinds of cellular differentiation earlier, I wanted to share with you one more example that's involved in muscle cell differentiation that I think is really interesting.
Ta tanda nga chu rigzi ge collations ta bala ya rigzi ge phont ge ngo lan jer bi kapse ane karsa rigzi de ta sko ra rigzi ge karsa shadring ge phont lan jer kapse rigzi ge ne ko chepar kari ju ge me de la shikta ge yes.
up at the top here, we see a picture of just one gene, and this gene is called alpha tropomyosin.
Tata gangla yo pate resistic res, kumila alpha tropomyosin resistic res.
And something that's really interesting about this gene is that actually many different mRNAs can be transcribed from just this one gene.
Ta dere kheba kari ore system balaya tanda ge rigzi kan chindila tene dete ane panchen RNA te ko phapsi chiu ka se panchen RNA rig mando mangpu chila phapsi chit ko res.
Only 3 of these mRNAs are shown here. There's actually several more. And since there are all of these different mRNAs, these are, of course, creating other different proteins as well.
Ta tanda de system bala de la ya nga su ge panchen RNA mando substance yos tina jige jena mangpu yo res wa. Ta ko panchen RNA mando system bala de to la tene deta tzi mando tungi yo res.
And this is interesting because sometimes when we're first learning genetics, we learn that, there'll be one gene and that will make one mRNA and that will make one protein. And that does happen sometimes. However, there are many cases such as this where actually, even from one gene, you can make many different mRNAs and proteins.
Ta de ko yang chembu chi yo pa ge mando chi yo pa ge jinsen ka system ba chirtang asu kuat kan des regla na rigzi kan chi ne ani pangcha ara ne kan chi phasu chi yo pa rig chi phasu chi yo pa la samlu ko yo res chirtang ge na tinda res imba na tandan de ge ni tang kapse rigzi kan chi ni is ko rigzi chi ne ani pangcha ara ne te ko mando mangpo ching du phasu chi yo res.
And in this case, that will be really important because if it's this yellow segment of the gene that's included, then it will signal to the cell to differentiate and become cardiac or skeletal muscle. But if the red segment is included, that means it's going to be smooth muscle, even though all these green segments are the same.
Ta tanda de la ya picha shapa imba na tanda gange te la ya sarpu yo pachito sarpu ko ge tumbu sarpu yo pachitang wala ya marpu yo pachito ka ke te gange tumbu sarpu yo pati sarpu yo pa imba na yang ko nga chu ge nyang ta ya na nga chu ge rje ge shatmul la ngu la chanju chi ta de te ge wala ya marpu yo pa imba na nga chu ge jamshe ngu la ni chanju chi ge yo res wa ta ko tanda de la ya chapro yo pati gange sarpu ni ja wo ke ngombo te ma to pe jenda ti ge jablo la yo pe ge jangko sama chi pa chi chang res.
So for time, I'll leave it there now. And after lunch, just so you know what to expect, I have a few very short videos to show you of the process of embryogenesis because I want to make sure that we're all able to see that it really is a process always in motion. It can be hard to tell that only from pictures. So we'll watch a couple of short videos and then we'll actually make a model of embryogenesis.
Ta tanda shopate to science ta nyingko jabla ya nga su ge dimsang driven ti ko la keche she pa imba na ko rumu khashi gang de she ja la khapu yo res tin stambala ya nyingko jabla ya ko ti ge taptang tan de wi ge lognin khashi chik tan yes lognin chongchong khashi chik tan yes ka chila pa imba na chirtang ge ko gen jiro do ya tanga jirum ti kanza mampu chi khuta do tan rojin ba stambala ya dundu ba stambala ya rumu kan tan khapu chik dus ta tin stambala ya ane lognin thong khashi chik ngu peche tang de tan ten nu ge jela ya ne ga su ge tanda ge ti tan drevi ge seguro ata ane basic zu ya chigoro wa oti tus nang yes.
Was there any question?
Try yes.
Okay, thank you. I know when it's lunchtime, no questions. No, enjoy your lunch.